Welcome to BioConcept, Learn with Concept YouTube channel. As we already know that, the adaptive immune response is divided into two types: humoral immune response and cell-mediated immune response. Cell-mediated immunity is particularly effective against intracellular pathogens, which include any viruses, bacteria, or fungi that are inside cells, some cancer cells, and foreign tissue transplants. The T cytotoxic cells, which have T cell receptor and CD8 marker on their cell surface, bind with infected cells or cancerous cells and distort them in two ways. Cytotoxic T cells kill infected target body cells much like natural killer cells do. T cytotoxic cell binds to antigen peptide with MHC class I molecule presented by infected cells, then undergo proliferation and differentiation into effector cells and memory cells. Effector cytotoxic T cell then releases granzymes, protein digesting enzymes that trigger apoptosis. Once the infected cell is destroyed, the released microbes are killed by phagocytes. Alternatively, cytotoxic T cells bind to infected body cells and release two proteins from their granules, perforin and granulosin. Perforin inserts into the plasma membrane of the target cell and creates channels in the membrane. As a result, extracellular fluid flows into the target cell and cytolysis. Cell bursting occurs. Other granules in cytotoxic T cells release granulosin, which enters through the channels and destroys the microbes by creating holes in their plasma membranes. Cytotoxic T cells may also destroy target cells by releasing a toxic molecule called lymphotoxin, which activates enzymes in the target cell. These enzymes cause the target cell's DNA to fragment, and the cell dies. T helper cells recruits other cells to infected area and activated them.